Okay, so little recap once again. So these last two lectures, I focused on what were these one locus models. That is, I said, you take this full genome, etc. I don't care the whole thing. Just replace that, whatever is important or useful for a question of interest by the whole chunk by a single letter A or A1 or A, and that could have two variants, A1 or A2. So that was a single locus biallelic model. Okay, so one locus. Why not two such regions, right? This chunk, and that chunk, etc. So one locus, two locus, three locus, and so on, right? Okay. So what I want to get to towards today is such multi-locus genetics, and that has connections to quantitative trait evolution when these chunks or these loci are very large. So I'll try to build up towards that, and let's see how it goes. Before I could get to it, I still need to spend a few minutes on just a two locus model, okay? Because I want to introduce something called recombination, which we haven't considered so far, and that's sort of useful what I want to say next. Okay. So let's continue along this simple models, the model simple kind, and this is probably the fourth or fifth one. So here, again, no drift, no selection, no mutation, but I just want to introduce this thing called recombination. Okay. No selection, no mutation, no drift. And I'll just consider a two locus model. So what I mean by that is, I have these two chunks, first chunk and second chunk. Each of them is biallelic. So at this first locus, so locus 1, locus 2, is biallelic. It could have two alleles, A1 or A2. Similarly, second one could have B1 or B2. So I have such 4 genotypes then, right? These are our genotypes. Okay. And again, I'm asking the same question as in the first lecture. What is the frequency of these guys? How does it change? What is the force here? I mean, it's not like force. It's if you like, it's from this last category I said, population structure. The structure here, it just means that these guys could have a genetic mixing. So what do I mean by that? Let me explain. So if I have these two genotypes, A1 and B1, and say A2, B2, what could happen is that they could break and then swap the genetic material at that locus. So that's what I mean by recombination here. Okay? So break and swap. A1, B2 could be produced, or A2, B1 with a certain probability, which is a recombination probability C. Okay? And I want to just write down equations about how these frequencies are going to change just because of this genetic mixing. Okay? So let's try this out. So just like we did for the Hardy-Weinberg case. I want to get A1, B1. So which genotype should mix to give me A1, B1? B2 and A2, B1. People agree? Yeah? Okay. So let's write that. Therefore, in discrete time, it is a recombination. Frequency is same. And because of recombination, there's a gain term. So I guess you heard that in the context of master equation. So there's a gain term. If I have these two types present, these two will recombine with a probability C. So A1, B2 has a fraction x2, A2, B1 has a fraction x3. So x2, x3, right? But, one sec, let me finish this. But A1, B1 could also recombine with which ones? Do you think which would change its frequency? Itself, if it does, no change occurs. It just remains A1, B1 with A1, B1. There's no change. Same, no? Which ones? Yeah. So that's a subtraction, x1, and then that's the x4. Yeah. So x2, one try, A1, B2, A1, B1. Break and swap, I still get the same guys. So they're not going to be useful, yeah. C is the recombination probability. Okay, right? Okay, so let's fill up this table a bit. So I need there's no mutation, no selection, nothing, just recombination. What did you say?
So
this is the gain. If these two recombine, and if A1, B1 recombines with A2, B2, there's a loss. Its frequency is smaller than what it started with.
right. So isn't there a flux of genotype A1, B1 out of the box?
Which box? I didn't get the question.
So you're considering the influx of.
This is a negative term, right?
Right, right. Okay. Okay, so C goes like that. You multiply the two.
Yeah, let me repeat this bit. So I'm saying this is A2, B2, A2, B1, break it, and, sorry, this is A1, B2, sorry. Yeah. Break it and swap the material. So I'll get A1, B1 produced. On the other hand, A1, B1, if it recombines with A2, B2, this will be lost because I don't, A1, B1, it will become the other two types.
Right.
Okay? Okay, so let's fill this table a bit and write down these equations. Okay, so you see a pattern, there is some quantity, x2, x3 minus x4, x4 in all of them, right? I could have just written a symbol for that, but let me do that now. So this object, x2, x3 minus x1, x4, it has a special name. It's called linkage disequilibrium. So I'll frequently write it as LD, or call LD, and denote it by D. Linkage disequilibrium. And as before, you can check, of course, that the total fraction is 1. Right? Okay. Okay, so what does it mean? What is the meaning of this bilinear object? So let's just do a little bit of small manipulation. So using this conservation, let me rewrite it as And okay. Yeah, it's okay. So this we know from the table is the frequency of A1, B2. What is this object? X1 plus X2. That's the frequency of A1, B1 plus frequency of A1, B2. That is the frequency of A1, allele A1, irrespective of what's the other one, right? So this object is f of A1, and similarly, this is f of B2, right? Okay? So now this is sort of easier to understand. What it's saying is LD is simply the frequency of this two locus object minus the ones when they're separated. Of frequency A1 alone, irrespective of who's the other one. By separated, I just mean irrespective of who's the other one, other locus, similarly times the frequency of B2. So first says this reminds of correlation functions. I mean, it's not real space or anything, but it's something like a correlation function that you encounter frequently in physics. Okay? Okay. So it's a measure of the linkage, because here the first term is saying A1 and B2 together minus frequency of this alone times frequency of the other one alone. Okay? So whenever we have this more than one locus of output, I have to worry about these objects called LD. And that actually complicates problems, but it also makes it interesting. Okay? So when D is 0, when LD is 0, things simplify somewhat, because it just I can write the frequency of this genotype with frequency of each allele separately. This, so I'm saying if there's no recombination, then what happens to frequency? There's no selection, there's no mutation, no nothing, there's no change, right? But now, because of recombination, this is the frequency. So what is the meaning of recombination once again? It's a, let me just write this one down. So recombination, I'm saying, break the two loci and swap the genetic material at the respective locus. We can write for all four of them, right? Yeah, okay? Okay, so how about this D? So because the frequencies are changing, the D would also change. So D prime, so prime, remember, is the one in next generation. X1 prime, sorry, X2 prime, prime. And if you plug in these equations and do a little bit of manipulation, you'll find that this is equal to, okay, it obeys a very simple equation. The LD is equal to 1 minus C times D, C is less than 1, okay? Therefore, D in generation P decreases geometrically, or, if you like, exponentially in time at a rate 1 by C. Okay? So why is that interesting and important? So it's saying over time scale, if the recombination rate is, or here it becomes rate, so if this rate is high, then the time taken to break this linkage, the togetherness, is small, it will quickly break away, and I have just these correlations I could ignore. So that's true, like for example, very highly recombining ones, like sexuals, right? But for S, which recombination rates are small, we have to worry about the full, the linkage is important. We have to worry about this full L locus problem. Okay? Yeah. Recombination rates are known. Yeah. There are numbers known for that. Okay? Okay, so that's what I wanted to say. Okay, so let me just say this. So it's decaying exponentially fast over a time period of 1 by c, and when d becomes 0, we call this term as linkage equilibrium. It's a very useful starting approximation in certain cases. Okay, so this I'll use this term, yeah. That's what I explained. The meaning of D is this. I mean, I chose A1, B2, but you can choose whatever you like, any of them. So it's saying that D is simply the frequency of A1, B1 minus frequency of A1 into frequency of B1. Okay? So the interpretation is of the genotype of 2 locus genotype minus the frequency of the 1 locus guy times the other locus. If linkage is it's like correlation functions. You learned that, right? Probability of A intersection B. So if the independent variables P of A into P of B, something like that when D is 0. So with time, if there's no other forces, LD decreases exponentially fast. Next generation, it's smaller by a factor 1 minus C. So, so it's just together thing. And then because of recombination, you have broken this part. And then it was like, this was my friend. He or she was always with me. But then I changed my friends. So, if I do it enough times, there's no associations. It's a measure of associations, if you like. Yeah. D0 means they will come together and go, but the association will be random because this guy comes with this one, so it's seen as frequently, A1 is seen as frequently with B1 or B2. So I will not see A1 and B2 together more often than A1 with B1 or B2. That's why I'm thinking of it as association or correlation. Yeah? Yeah, so I mean, it's just saying it will be just like as if they are randomly, so this is a measure of saying that the association is more than if they were distributed randomly. More than, this is a non-random association, that's a correlation between them. I mean, I still have a 2 locus problem. It is always a 2 locus. But what I'm saying is A1 would be seen if I just sample it. If there's no linkage disequilibrium, if there's no LD, they will be associated with each other quite randomly. So think of it asexual, A1, B2 if I start, it will remain A1, B2. A1 will always come with B2. Because of, recombination, it can come with B1 as well. Think of it, these 2 limits, asexuals. There's no recombination. You always stay with the same friend for all time to come, right? Try either, same. Yes, but that's, yeah, right. That's. No, that's simply saying that x1 prime plus x2 prime equals x1 plus x2 means the frequency of B1 plus B2 plus frequency of B2 it doesn't change. So that allele, yeah, that's a marginal, right? Yeah, yeah. Yeah, this like a, yeah. Right, that's the definition of FA1, no? Yeah, yeah? Okay, so I want to leave, okay?
Isn't the distance between the locus considered here?
No, I've not considered, and I should really worry about that because, the ones which are close by, they should have low recombination. The far off has high recombination rates. But I'm just considering 2 loci, there's nothing else to compare with.
Okay.
Right, so C is variable within a genome, right? Yeah. So that's what I'm saying. If it is low, that means they are probably also physically linked. Where C is large, they are far apart. But if I, fix these loci, I'll have a fixed C value. Okay. Okay, so I want to leave this guy because I just want to introduce this because I want to use this later on. And I just want to start with this full multi-locus genetics. So we started with one locus, and I want to now consider L loci. And this is something I'm learning myself. So it's quite possible that I don't know the answers to all your questions, but I'll try it out in you and see how it works. Okay? So let me just begin with a bit of overview of things a little bit, because that's something that worried me for the last few years, and let's see if I understand this a bit better now. Let me start. Let's leave all this and take a slightly broad view of things. Let me talk to you about theories of adaptation. So adaptation, everybody has some understanding of it. So you can find lots of reviews, and one person I would recommend strongly is to read these reviews by this guy called Alan Orr. He writes very nicely. Reviews, on various subjects, but for example, this is also nice to see for this in this topic. Awesome. Okay. Now, these theories of adaptation, they are coming, they work at different levels. One of them is like, they are just defined only in the phenotypic space. So by now, all of you know what's phenotype, right? Okay. So you don't care about what's happening at the genetic level. You just look at how height is changing, how the Drosophila's, wing or something like that is changing with time, etc. Right? And there are a lot of bunch of models. Some of them are like Fisher's geometric model, which I think we won't discuss in these weeks. The entire field of quantitative genetics. Okay. And that's fine, because, selection acts on phenotype, and you just worry about phenotypic level. But, we know in these last 100 years, we have learned a lot about what happens at the genetic level. We would like to know about that. So that's what I'm not so happy with this. What's happening at genetic level? Right? Okay. Then there are models that are defined purely in genotypic space. There are lots of them again. Let me just mention a few. Adaptive walks, which is like saying there's a sort of fitness landscape, which is like a very high dimensional version of, if you like, Himalayas, and you have to climb uphill, and under certain conditions, it's pretty good, but not good enough. And there are more sophisticated versions of that, and those theories are called clonal interference, etc. And I think Luca will probably do that a bit today. There are lots of them. I just mentioned a few of them because they relate to adaptation process, right? Okay. But here, the problem, which is not nice here, is that because selection acts on phenotype, and when I'm looking at the genotypic level, I just say that what I should really do is to define fitness in the phenotypic space, go from phenotype to genotype, and therefore find fitness in the genotypic space. So you need to go through this route. And when I started out as a physicist, it was like, I don't know how to do this mapping, and not much was known either. So typical approaches, let's forget about all that and specify selection, all these numbers that I have been writing, 1, 1 plus, etc., directly to the genetics, to the genotype. Okay, so here it specifies selection, without any regard to phenotype. just like this does not address what happens at genetic level, these models generally don't care about what happens at genetic level. So therefore, of course, I mean, the obvious thing to do is to link them somehow, right? So, link phenotype, genotypic, genotype evolution. And this is not something, this great idea that has occurred to me today. There is a long tradition in population genetics that worries about this question about how to understand phenotypic evolution, quantitative evolution, starting from genetics. And at least since 1960s or so, probably even before. Okay, this is not something new. But progress has been slow because these models, as you can imagine, are pretty complicated. They're technically more involved. So progress is slow, but that doesn't stop us from trying, right? Okay, and there are also a bunch of models that I just mentioned because you might encounter them. So several of them. So there is this model called infinitesimal model. I'll say a bit about that today. Then there are this thing called infinite alleles or continuum of alleles model. Slightly different continuum of alleles actually. Then there are also sort of finite alleles, multilocus models, and there's a whole bunch of that. Okay, fine. Okay, so I would like to carry out this program. That one. So infinitesimal, this relates to this. I'll say that later. Okay. Okay, so I would like to carry out this program. I mean, in this traditionally, like the people have done. And as I said, it's difficult, it's challenging. So I'll make lots of assumptions, approximations, but we'll make perhaps a bit of progress. We will understand things a little bit better. But then it also opens up lots of new questions, which people could try to address. Okay? So let me start from some basic things, and let's see how it goes. So I said the proper thing to do would be to look at phenotypic fitness, have a phenotype-genotype map, and therefore arrive at the genotypic fitness. So there are some, not understanding it because you don't go and actually measure fitnesses. There's some proxies that okay. So you do measure fitnesses of a phenotype, with whatever data you have, and you sort of make some simple fits to it, etc. And then on that basis, there are some models for this. I'll just mention it to you, and some of them I'll use. So at that time I'll define. Truncation selection, stabilizing selection, directional selection, and so on. Balancing selection. Okay. When I come to them, I'll explain. Okay? Okay. But let me talk about this guy first. So, this is something I guess you have seen before. Have you? Right? I like Z because P is probability. Okay? So, but this is the sort of standard notation in the subject also, so I'm just going to use Z equal to G plus E. Okay? So, this is the phenotypic value. It's called a genotypic value, and this is something to do with the environment. So what one says here is, and I think you heard already first day about these things, so I'll just say a little about this. But the point here is the following. Suppose I have a certain sequence. So you imagine doing an experiment in which you take genetically identical organisms, individuals, and rear them in a large number of environments, infinite number of environments. So let's say you take everybody with a certain sequence A, okay, and put them in different diets, some of them will exercise, some don't, etc., and find out the distribution of heights. So prob height, okay, let's call it Z, is something like this for that sequence. Take a different sequence, do this experiment, let's say TGTTT, right? If I repeat this experiment, I'll probably find a different distribution. Maybe. So this here, the variable is the environment. Okay? And
I'm sorry, just to be clear, so Z is?
That's the phenotypic value, that's the height.
Of the height. Yeah. And
of
then
all the individual which have the same, which are genetically identical.
Okay.
They are reared in different environments, and that's why I have a distribution.
Right. So, you fix your genotypic value A.
No, I don't fix a genotypic value. I fix a genotype. Yes.
A genotype. Yeah.
Yeah.
And then you Z goes over environments, and then you
Z
see
is the trait value, which is like my height. I mean the height of those individuals. So you imagine. You take lots of individuals of the same genetics, the same genetical sequence, give them different foods, different exercise, etc. Okay? So there will be a distribution of heights, I'm saying. Okay? So this average of this is called the G, or the genotypic value. Okay? So this, of course, depends upon the sequence. This is the object which is averaged over the environments. That's what G is. Okay? So that's my G. So therefore, if I look at one individual in his cohort, then he or she would have a height different from this average. So then I'll say that that's the environmental influence. So Z minus G of that individual. Z is the height of the individual minus the number that you got by averaging with environments. That's the environmental component. Okay, so that's what you have already seen. Okay, so let's just look at this guy. Okay, so that's our Z, G, and E. Let me say a bit more about this G. So what is this G? So I said it's a genotypic value, it's a number, fine. How does it relate to the sequence? Yeah. We would like, because it makes life easy. Typically, normal is what is considered, and if you change certain scales, it looks pretty normal. Okay, so what is this G? Now let me not say it's a number, etc. It depends upon the sequence. And now here comes the map. So I'll assume for today's talk an additive map. So what does it mean? Let me explain. So I'm thinking of a sequence, sigma, which is composed of L letters. Okay, sigma 1 could be 4, it could be 2, depending upon what sort of thing you're interested in. Okay, okay, fine. So these are, let's say, Biallelic, or, okay, I'll make this distinction a little later. Okay, so then my genotype G, I write as so here, this is the effect of having, so suppose I had, okay, let me explain this better. So suppose I had a sequence like this, A, T, C, A, okay, let's say L is 4. If I had an A, let's say it increases the height by 5 centimeters. If it has T, then maybe it decreases the height by 2 centimeters or something like that, right? So by the G, I mean that whatever is the state and its effect. That's what I'm denoting by Xi. Okay? Which one? Additive maps? Additive? Additive maps, is that what you mean? Yeah. So first of all, I would say it's a null model. It's a null model. I would think of it as a null model, right? Because it's easier. Not so easy actually. I'll work with only this and then you will see it's still complicated. Okay? So it's deployed, so mom, dad. So I'm sorry, notation is changed simply because notes are written at different times. Okay? But yes, I should have. So this is what I mean by additive. That means the effect of each locus, each nucleotide. So I just add it up. This moms, this dads. Plus, it should have other terms. Yes, xi, xj, xi, xj star, and y stop at 2, 3, and so on. So what that means is, if I had an A here and T here, the effect is different from when I had an A here and an A here. So those kind of interactions, that's the epistatic interactions. So those terms should also be there. And they do occur, data suggests that they are there. I mean, I'm neglecting it simply because they are difficult, and I mean, I don't know enough about it, but there is a lot of work going on, and you should look at those recent papers. Yeah, this is all some function, which I don't know right now. Okay?
Kavita, I'm sorry, but could you say what's the star in there?
This term?
This star. Xi?
Star is, this is the effect because of mom, and this is because of dad.
Okay.
Okay, so I'm sorry, this is changed. Okay, I'm just following. Okay, I should just say. Okay, let me just stick with this. Okay, so this is what I'm going to neglect today. And they are important, etc., but we'll do just this guy. So, okay, so this is, then Z is equal to this one plus the environment. Okay, and I should just mention here, experimentally, what do we know about these maps, etc.? So there are definitely departures from additivity. They also depend upon environment. So if you do, figure out these effects, etc., in one environment, it's different in the second environment. So it is very complicated. And let me just mention this review. It's a bit old, but it's quite nice. So this Trudy Mackay, she is a big expert. So this is in, I think, Nature Reviews Genetics. Okay, so if you want to know about what's normal experimental, what's the patterns, trends, etc., that are known. Okay, so we'll stick with this genotype-phenotype map, Z equal to this plus an E. Let me now say something about E. E is a random variable. As I said, eat less food or give different, less food, more food, etc. So these are these environments, and they are typically assumed to be this random variable E to be normally distributed. So the variance here, and we don't expect usually that if I have infinite number of environments, I don't expect one environment to be, an environmental bias if I have infinite number of environments. Therefore, it's a distribution which is normal with zero mean, and the variance being V. Okay? Right. Fine. So this is a little setup. Yeah. So what you actually want to measure is these effects. Yeah. So X means that for the sequence. Yeah. So, but yeah, the effects is what you can measure. But the data is very less, etc. So what you do is to find out, okay, if I put ultraviolet light, did the corn stalk increased or decreased? Yeah, so that is the problem. I mean, we can't really for this. So what one does is to say, okay, it's a very large number of loci. Let me just, look at all environments and just draw a distribution. Doesn't matter whether it was from ultraviolet case or, more humidity, etc. And just think of it as a random variables, the effects. So that's a useful way of thinking. The effect sizes, they are random variables. I will not do that. I will assume today they are constants. And same all for each locus, so whether you're A, T, it's same because it's a simpler model. If you want to consider different effects, there's recent work, you can have a look. So there is some progress, but let's not talk about that today. Okay, so this is a little setup, and let's just do some simple exercises because I need them later, and also it will help you to get used to it. So we'll work with this guy, and I'll take a simple model. Of Biallelic. Let me explain. So what I mean is xi, if it is, let's say, is A, if that sequence sigma is 1, if it's minus 1 or 0, let's say, then the effect is minus A. So I'm saying I have a sequence 0, 0, 0, 0, 1, let's say, right? So the xi would be A, contribution from A here, so minus A, A. That's what I'm by summing over all the loci. So summation xi for mom, that will be for this sequence minus A plus A. Okay? Okay, so why don't you guys help me with this a bit? Let's calculate a bunch of simple things. I want to calculate Z bar. Phenotypic average over genotypes as well as environment. So that's equal to G bar. This is averaged over the genotypic frequencies. That's what I mean by bar, plus E. This is the average with respect to environment, with respect to genotypic frequencies, and this involves both the parts. And because I said average is 0, it's a normal distribution, this term is absolute. Okay, so what do you think this object is? For that map, xi plus xi star. I just give you this example when xi is A or minus A. So I look through this large population. I forgot to say, I'm going to assume now today, no drift. It's a huge, deterministically large population. Okay? So if I have sigma, wherever I have 1, okay, because I'm averaging it. So this would be 2. So wherever you have A, you write an A there, effect, and the frequencies of A, the 1 is P, and that of minus A is Q. Is this okay with people? And 2 because it's diploid, and L because the L such loci. Yeah, makes sense?
It is measuring both contributions of father and mother.
That's why I put a factor of 2 for diploids. A is same for mother and father.
Okay,
Yeah?
okay.
Today we'll just keep it same all through. Is this okay? I want you to try this. If this is okay, how about, that's a mean genotype and mean phenotype value. Let's just make sure that we understand. Could you calculate this object for me? This variance. Can you just try? It will help me to know whether I'm getting my point across or not. What do you think is this term? I mean, it's not 0 or anything. You have to actually work it. But just give me some idea that what would it be? This is something you have seen, I think, in this random walk lectures or something like that. Right? It's just the variance calculation. Yeah, right. So it's just that, yeah. Right, very good. But you have to worry about the frequencies of each of them is IAD. That's the frequency. And I'll just give you the answer. Very nice. 8 because of the 2 factor of diploids, etc. P cube LA square. Okay, please check it. Okay, but I want to use these little formulas. Okay, so this is LA. I'm sorry, I'm missing sometimes these factors of 2. They're not so important today. Okay. I added this subscript, sigma A square. This is the variance in the genotype, and because I assumed an additive map, I should have had all those terms. So to distinguish that, that's why I'm saying this is called an additive genetic variance. Okay, so fine. So I'll just do this one last bit, and then I want to relate it to some of the things that you have seen in other lectures. So we have some small understanding of what this map is. So G is, you can expand in terms of these the sequences. Then I said I should start from there, and I should start from phenotypic fitness and relate it to genotypic fitness. So let's do a little example of that. This is one, little example to see that how to calculate these means and variances. I need them later on, and that's what you typically calculate. Okay. So if you like, this is a little second exercise. If I'm giving you, I want to calculate fitness of a genotype. Okay. So that's equal to. So since you know central limit theorem, that's nothing but make sense? Z equal to G plus E, and I'm averaging with the environments, right? That's what G value is, and I know prob of E. That's what you write now when you do central limit theorem, right? Okay, so that's this object, and you can calculate it for different cases. I'll just give you the examples, the answers here. There's something called directional selection. So where this WZ is on a log scale is linear in Z, and therefore you can just do this integral. Turns out to be some factors. It's just a matter of doing this little integral. Okay.
Kavita, sorry. What is W? Please come. Could you give us the intuition for WG? What's that integral? What's that?
This is the fitness of the phenotype.
W is the fitness of the phenotype. Of the genotype.
I'm getting it now starting from phenotypic fitness. So suppose the phenotypic fitness of this type. Okay, it's e to the power Z. That's what is called directional selection. That is the tendency to increase that phenotypic value.
Right.
Linearly increasing.
Right.
Scale.
Right.
Right. So then, using Z equal to G plus E, I'm deducing what is the fitness of the genotype.
Okay.
So I'm not just ascribing the value, simply forgetting what phenotype. Here I'm doing it systematically.
Okay.
Okay? Yeah. Let me just give you one more example, because you've seen it in other lectures. So that is the stabilizing selection. So here, WZ is a Gaussian, centered about some optimum value, and there is certain width to it. Right? So I think you saw that, right? Like, for example, the baby's birth weight. There's a tendency not to have too high birth weight or too low birth weight. So birth weight is a phenotype, and it is centered about some optimal value, and that optimal value is Z naught, and the width is omega. So that's what you're getting from experiments, for instance, and then using this little integral, you get WG. That also turns out to be Gaussian, but with a different variance. So this is a set of written examples to tell you how I said start from the phenotypic map, use Z equal G plus E, and therefore reduce W of G. Okay, so this you can easily work out, etc. Okay, but I want to make a little point here. If I assume this Vs, this is the strength of the stabilizing selection. So if Vs is large, that means the all kinds of phenotypes are, okay. Their strength is not too, they are okay. Their fitness is not too low. So then we say selection is weak. If it's quite sharp, then we say selection is strong. Typically, we take Vs to be large because for theoretical reasons, and therefore I can expand it. And let me, when I do this thing, and I take W bar. So I'm giving lots of answers here, simply because there's less time, etc. But I'm here, so you can always talk to me later. Okay, so I just expand this guy and take some expectation values. So that is equal to so these are very straightforward exercises. Okay, because I'm ignoring higher order terms. This is the quantity that I mentioned over there, sigma square, for the additive map. And this is for the additive map, Z bar minus Z naught. So it's about the deviation of the phenotype from an optimum value. So it's some formula, etc. But the point I want to make here is the following, because I need that later. The fitness, average fitness, is a function of mean phenotype, variance phenotype, etc. Okay, so that's the key point I want to make here. This is genetic variance, but Z equal to G plus E, you can check it, means that sigma P square or sigma Z square is equal to plus other interactions, which I'll neglect today. Okay, so the key point, let me just finish this. I just want to get to a point, that's why. So the key point I want to make here is that W bar is useful to think of it as a function of mean, variance, covariance, etc. of genotypic values, or of the, in this case, phenotypic values as well. That's one thing that I want to use in a minute. Yes. Z is equal to G plus, okay, what did I do? Yes, so what did I do here? Yeah, so this will become W G plus E P E P E. So Z I write, so okay, suppose you want to keep the Z, you change the variables back to G, this is what I mean. Z is equal to G plus E, so this is integral over E, where this W is this guy. Okay. It will click only there, that's why. Yeah, so if I expand it and take average over genotypics, so then you will see that it has, to this linear order also, two components. It has a variance component and the mean component. Yeah. Okay, I understand.
W is the distribution of Z.
I should use.
This is distribution, no?
Yeah.
And the second, okay, and the second question is probability of E. Means you are finding the probability of finding that particular environment. Means
probability that, so I don't know, environments are lots of them. I mean, I'm not, there's generally access, right?
Yeah.
I'm saying there's somehow I can reduce to a single number, E, a random variable E.
then how to get that environment probability? Means it's very.
Yeah. No, this is the probability distribution, the E, the probability of E. W is the fitness, yeah.
W is the fitness.
Yeah.
Then Z is?
That is the phenotype.
Phenotype value. So W is the fitness.
W is the fitness.
Okay,
of the phenotype.
okay. And the second question, probability of E. So one particular, how to find a particular environment probability means just in a practical purpose.
I think it's just an assumption saying that there are lots of them and it's a random variable, perhaps composed of lots of variables, maybe from central limit theorem.
Exactly.
Because this is trying to stabilize things towards intermediate values. If you go away from this, you tend to die and you stay here, that's a stabilizing. And this one is linearly increasing fraction, like a velocity.
And that fitness value even,
Yeah.
that fitness value of the genotype, is that also assumption or derived? Means the WZ you started with?
This?
Yeah, derived?
There is some empirical work on this, like a birth weight of children, right? But infant child, the birth weight. So that has some, shape like this.
I just want to recall, maybe you were at the ecologist lecture we had from a back that we saw those level curves.
Yeah.
That seems to be an stabilizing selection, right?
Yeah. So that's the same. That's what I'm mentioning here. It's a stabilizing selection. The birth weight is one example of stabilizing selection. But lots of traits are under stabilizing selection. So it's worth modeling it and working it out. Yeah. I'm just modeling it, saying it's in a direction that if you increase, like for example, you just increase the height. You are under such a environment that the height should increase. But increasing height is better. Linear things are easy. Log scale linear is easier. The one reason the stabilizing selection, a lot of work on it, is simply because it's quadratic. And that is complicated. I mean, I won't be able to explain that to you, but this just making it quadratic makes things very difficult. No, that's easy. So that should be more acceptable. Okay, so, yeah. It's over the frequency of the genotypes, which I want to come to. Should I do that? Okay. Yeah. Not that I know of. I mean, you just do this guy. There's not too much experimental work, or even, for these things. See, the typical thing is this. So you make a model, and then you see whether it works, meaning, from experimental. If it's consistent, at least maybe there's something in it. Besides that, you could make this as Z, maybe make it root Z, Z square, if you like. If it changes the behavior, yeah, should put it in a different box. Right? Everything is not like, just put experimental data on top of it. That's not modeling. Yeah. Genetic invariance has different components, additive one, epistatic one, and there are other terms like dominance, etc. All that I have thrown out today. Because my genotype is simply the addition of effects at each locus. I'm not allowing them to interact, etc. Right?
Ma'am, hello. the W.
Yeah, I think I'll just go ahead because
Yes,
I want
ma'am,
to really
one second. Just WZ expression. The two W in the both sides are same. Means that's standard deviation W and that W are same. WZ expression.
Yeah, this is the.
Not that expression, WZ, e to the power minus of that expression.
Yeah.
There is one W in the denominator.
This is omega.
Omega, okay, they're different. No? Okay.
Yeah, omega is different from W. Okay, so please, I really wanted to go to this thing called Breeder's equation, which you heard from quantitative genetics talks, etc. I really wanted to derive it under some bunch of assumptions. So time is short, so why don't I just go ahead and I'll take the questions later. Good. I mean, it's rather involved, etc. And it'll take some more time to answer all the questions. Okay, so let me set up this model for multilocus case, and I'll consider only selection. And there's these references. That basically I'm going to follow. They have these titles like Multilocus made simple, and you'll see how simple that really is. At some point, Landy, 76. Okay, so the setup is from these guys, and then I'll use some assumptions of Landy. I mean, he has also a lot of work on this. Okay. So let's write down only selection and also recombination. No mutation, no drift. Okay. So that's the sort of species I'm thinking of. So f of x vector denotes the frequency or the distribution of these states that I said. X1, it could be 1 or 0, it could be many alleles, etc. So it's just the state at the first locus, second locus, and so on. Okay, that's what it denotes. And I'm going to drop the x star, but x means x and x star, both of them. But I'm not going to write it today. Okay, so this we have seen. So now everything is at genotypic level. Okay, I'm going to come back to the genetic level. I will work there, and I'll try to go back and derive this thing called Breeder's equation, which is at the phenotypic level. So I want to go through this multi-locus route. This you saw the first lecture, right? Therefore, rate of change, the change in one generation. Okay, fine. Where W bar. I'm going to rewrite this bit. I'm going to divide some things, multiply something, etc. Okay, so but you can check. So I'm going to write this guy as so dw bar or df with respect to some other state is equal to wy vector, right? So I'm going to use that and replace this w. So I have okay, and now Because you know this is W and this is F, it's W bar. This is W bar. This is the W bar here, no? Oh, okay. Yeah, thanks. Right. Okay. So I just, using this, I but putting this guy, and this you can just check. There's a delta. So it's just rewriting this guy so that I want to get this d by df of log w bar common outside I want to keep. That's why I divided, multiplied, something. It's sort of easy. It's not so difficult. Okay, you could check it. Okay. So once you are ready with this, then let me write down the mean value change. So mi the expectation value of xi with respect to this distribution f of x vector. So therefore, this change at the ith locus using this is simply I pointed out to you that it's better to think of the mean fitness not as a function of these, of course it's a function of all these frequencies at each locus, right? It's better to think of it as as a function of these variances, covariance, etc., etc. That's what I'm going to use now. So it's better to think W bar to be a function of, that is at Mi, which are defined, such variances, let's call it Cij, variance, covariance, etc., third, cumulant, and so on. Okay, if you do that, you do some change of variables. So you write this guy as similarly for the covariance term. It includes also variance. Okay, so it continues, for lots of terms. Use this guy, plug it in here. Then, okay, so let me drop these terms right now. Why, etc., I can explain later. Okay, let me drop them. We can add it. It's not a big problem, but it's just that it will make my equations very long, and I don't need it for now. Okay, so I use this guy, plug it in here, and work out things a bit. If I do all that, yeah, I know. I just wanted to mention what all I need to use. So I need to use this guy, and I am also using, because Z bar is equal to G bar, therefore, is equal to, I'm going to use this guy and this one. This will allow me to take this term outside the summation. Then I have a summation over J, and then I have to do, I'm sorry, I forgot to, yeah, no, I have everything, yeah. So I'm going to take this term, because of this, outside the summation, and just put things together, okay? I do all that. Yeah, I think people won't be able to see, so it's okay. Okay, so let me just give you the answer. So you should get I have a summation over I also. I already had a summation over j because of the delta. Okay. And so on includes those terms that I dropped for now. We can argue that they can be dropped under certain conditions. Okay, but let's just stick with this guy. So we have, therefore, the rate of change, because delta mi means because of z bar equal to g bar, that's nothing but the change in the phenotype. So I said z bar equal to g bar, right? This object is precisely my rate of change, the change in the phenotype. So let me just summarize this. So if I follow these steps, I should get, sorry, I'm missing some factors of 2, but I should have something like this. Okay, I wrote it already. Or let's call it Cij. Okay. Right now I want to focus on this little bit only. Okay. Now I'm going to make some approximations, some assumptions, and let's see where we go. Okay, first thing I want to mention here is, I said these problems are technically difficult. If you have some experience with StatMech problems, statistical mechanics problems, you see that problem right here. I started to write an equation for the mean, and it's related to the covariance or the variance, that is the higher moment, higher cumulant. That's a typical thing that happens in statistical physics, and we are quite used to that. And then basically, we wind up with the same problem when we have these multi-locus problems. It's like a multi-site problem if you have a physics background. And then you have to work out mean field theories and other assumptions, approximation, etc. So that's where a physicist can contribute because you have already some experience with these things. Okay, so but this is a typical feature. Equations don't close. Let's say a sort of problem with these problem equations. Moment equations don't close. Let's see if I can derive Breeder's equation starting from here. Okay, so let me just now just remind you what that was. It says the change in the mean phenotype, which is also written as R sometimes, is equal to heritability h square times selection, where the selection is equal to z bar w at the same instant t minus z bar. This is called selection differential. So what this means is the mean phenotype today, but then to form the next generation, I don't use all of the individuals in the population. I select some of them. So that mean phenotype of the selected ones is z by w. So think of, again, let's say the cattle breeding or something like that. So you have P of Z of some kind, okay? So you say that, so this is the P of Z, which is usually assumed to be normal, simply because we're thinking of a quantitative trait which is supposed to be composed of lots of loci. And from central limit theorem, it follows that P of Z should be normal, okay? So here, Z bar means that, these cattle, they have certain characteristics, certain trait, and I want to use only this many, the top 10%, 5% or something. What I'm drawing here is the step function. If I use this as my fitness function for the phenotype, that's called truncation selection. You need to use that, but I just wanted to explain to you what I mean by Z by W. So this is an example of that. Okay. So you take top something, and so the mean phenotype of this population is Z bar W, and that you take to form the new generation. So that's just how we can get this. Generally, it's something derived. I mean, it's just something seen in experiments, a phenomenological equation. So let's see if we can derive it. So this is what I want to get to. I have the same left-hand side. Right-hand side is complicated. Let me make some assumptions and see if we get it. Let me just focus on the right-hand side. Okay, so here are the assumptions. So Z bar W, so the ZWT is just like we did for phenotype. The same equation, you have seen this like many times now. Therefore, if I assume that P of ZT, the phenotypic distribution is normal, which is okay, I think usually to assume. So this is a normal distribution. Here the variance is sigma P square, or in my notation it would have been that square, but let me stick with the standard notation. So this is the phenotypic variance. So then you can see and this is my P of ZT. So this is not dependent, this depends upon Z bar. I take the derivative inside and use the normal case, and this turns out to be yeah. Therefore, this is nothing but okay. So I'm trying to create that object that I want to prove. I want to get this guy. So this I already wrote an equation for, starting from our population genetics. I wanted to create this object, Z bar W minus Z bar, okay, which is what I have done here. So I note that dW by dZ bar is equal to this object, right-hand side of the Breeder's equation. Now, we're almost there, because you see, I use this, I have it's almost looking like Breeder's equation, right? So the derivative of the log W bar is equal to this object. For any W, I just gave truncation selection as an example, but this is for arbitrary W's, arbitrary truncation selection, stabilization, whatever you like. Okay? Now I still have a little bit of this guy. Who is this guy now? Okay, more approximations. Okay. So first thing I'm going to do is to assume, okay, I lost track of the number now. Assume what I mentioned at the beginning of this lecture. I'm going to assume linkage equilibrium. So what was the meaning of linkage equilibrium? The frequency of the together guy equals the frequency of each separately. Therefore, the terms, covariance terms would become 0, and my right hand side equals this. Somewhat like a variance, but just from few more steps. I should just mention here that if you don't like the fact that I have assumed linkage equilibrium, you could set up a perturbation theory, and you could add terms of the order S is some selection, some parameter of selection, time divided by R. So that can be done. So I will not even do that. And that kind of thing is called quasi-linkage equilibrium, QLE. But let's not bother about it today. Okay, so let's focus again on this. And I need to still worry about this. Okay, so the problem here is that the CII, the variance, that is time dependent. When you write Breeder's equation, usually this object called h square, it's not time, it's a constant, it's fixed in time. Under a model, which I can't explain now, called infinitesimal model, it is possible to argue that the variance will not change with time. If I have a very long loci, the number of loci that are contributing are infinitely large, and that A, the effect of each locus, is infinitesimally small. So I'll just tell you the answer here. So if I assume, let me just write it there. So, maybe third. So much of quantitative genetics is based on this model due to Fisher. And later on, there's a lot of work by Bolmer. So this says that the effects that I assumed, the sizes should go like 1 by square root of L. Okay, so that L goes to infinity, it goes to 0. And so one is thinking of a quantitative trait to be composed of a large number of loci, and each locus has a small effect. Okay, so in that case, it turns out, it's easy to show from the little equations that I wrote earlier, that CIIT is constant in time. Okay, so that's it. And this is nothing but my additive genetic variance. That's all I wanted to show. So we have the Breeder's equation, therefore. The numerator is now sigma A square by sigma P square times. This is your selection differential. This is your response. And this is the narrow sense heritability, it's called. The ratio of the two variances. And because it's variance is always positive, one writes it at h square. Okay? And sigma p square was nothing but sigma a square plus sigma e square. So this says the fraction of, the fraction that's inherited. Not everything, not the whole part of the trait is inherited. Only part of it is inherited through genetics. So that's measured by this thing called heritability. Okay, so I think I've run out of time. And yeah, so I think I should just stop here. Yeah. So, but that's a linear regression. That's what one does by fitting the data. And one finds out this h squared experimentally. But like, why?
I've got two questions. First, the two loci. The first thing that we started with, two loci problem. The self-recombination, the point that I gave, A1, B1, A1, B1, recombining with A1, B1, producing the same. So we did not take this in account. But self-recombination rate is going to make some cost. And should not they be incorporated in fitness or in the frequency mapping? My first question.
So if I had done this full thing properly, I would have a term. I already have selection. I would put a term. So I'll have a model with recombination and selection. So yes, we would be putting that, but I just didn't do it today because it is very involved.
It should come.
Yeah, you turn on one by one or all of them.
Okay. And the second question is about WG integration over WZ, a probability of that equation. One phenotype may result from 4, 5, or 6 genotype. Am I right?
Yes, if the effects are different, it's possible. Genotype to phenotype mapping could be many to one, yes.
Exactly. In the case of many to one mapping, the WG you were pointing out, which WG? Suppose there are 6 genotype for a particular phenotype.
I think today I'm thinking of only these additive ones and also say constant effect.
Means just one-to-one mapping, I may say,
No,
in that
not
case.
one-to-one. So what will it be? So it will be from many genotypes to the same phenotype.
Yeah, but what then WG is? Means WZ, one
So
phenotype.
many genotypes can have the same value. That's neutral.
So they are same value. It's a neutral.
It could be neutral, yes.
Okay. And the third question is that the construction of WZ, you said, I know that omega is the standard deviation for that case,
Yes.
for the stability case.
Stabilizing selection. Yes.
Okay. Then we are considering a continuous phenotype space. Am I right?
I'm
Okay.
considering a continuous trait, yes.
Yeah, but in most of the cases or in practical examples, for example, suppose the tall, short that you were telling, height, some phenotype space may be discrete, maybe 6 or 8 phenotype space. I have only just data or number there. No? I do not know the relation, functional relation. Even I cannot predict from 8 or 10 data.
If I have a discrete phenotype, I think they are generally controlled by very few loci. Like for Mendel's example, actually. The Mendel's peak is, I think, is just one locus or 1 or 2 loci that control it. So then the P equals to G. I mean, there's no really anything to worry about. So I'll just say A1. A1 is the wrinkled P. That's it. There are very few loci controlling the discrete trait.
But for that case, WG, in place of integration, there will be summation. Na?
Whole thing, yeah, you can do that. That's right. Yeah, we'll do a summation then. But this whole thing is really when I have a trait is controlled by a large number of loci. And that's why everything is continuous, etc. That's the inherent assumption. I should have said that before. Yeah. Yeah.
Yes, Kavita. So I was lost in calculations.
Okay.
So I want to take home like some words of what you just did. So how could you
So,
like very brief?
yeah, so I'll just tell you, actually I just wanted to say that, this worried me a lot. That why am I only in the genetic space? What happens in the phenotypic space? And when I heard this talks about quantitative genetics, what's happening at the genetic level? That's the overview I wanted to give. But the point I want to make, I wanted to make is that you can connect, you can make a bridge between phenotype and genotype. So you start from these microscopic models, if you like. As a physicist, I like to think of it that way. And derive these macroscopic things, like, about a phenotype. And that, lets me sleep well in the night. So, I mean, I feel better that way. And of course, it also elucidates what are the assumptions that we have made. I made a lot of assumptions, dropped lots of terms, etc. So, for example, Breeder's equation will change, it doesn't hold, if I allow LD. If I take LD into account, it doesn't hold. There are important differences. So this shows you, if you start from scratch, derive something, it tells you what's going in and start questioning that. It's not just an empirical thing, it's a derivation. So that's something which you are interested in. So I'll just close this by saying there are lots of questions. So for example, here this sigma A square is from environment. A sigma A square is a genetics, right? So this we should be able to derive from population genetics models. That's something people have been doing it for, again, from starting from Kimura, and lots of people have done that. You can ask questions about adaptation. Breeder's equation, etc. hold only for short intervals of time, few generations only. But I'm interested in adaptation for many, many generations that can take. Then you could use this machinery to answer those questions. So my student Archana, she has a poster on that. So I hope she can explain better than me to you. So do visit if this interests you. Okay? Hungry? Yeah. That was the variance. Xi, xj minus, so. Yeah? See, this is very difficult. Even my notes are not very good. So, but try these guys. I spent a lot of time. Their papers are extremely hard to read. If I can clear up my notes a bit more, I can share. It's not a problem.
Yeah, there's.
But try this guy, Landy, 1976. It's not a lot of calculation, but you have to buy some of the things, and it also relates to experiments. It's a very nice paper. These are much more technical. Once you've got used to some PopGen, you should be able to tackle them. Yeah.
Any book can be recommended?
Yeah, sorry. So this is, lots of people have been asking. Let me give some references, and of course, this is my bias, because that's how I have learned the subject. So if you are the biotypes, meaning you like biology more, comfortable with that. Less calculations, so there's a book by Charlesworth and Charlesworth. And there's another book by Barton and many others. I forgot the names of others. So both the books, so this book actually has no equations, but there's a nice set of exercises, etc., that you could look up. Charlesworth and Charlesworth talks in the main text about biological problems in verbally, but they have boxes which do some amount of math. Okay? If you are more into math, then a couple of books. One is by Ewells, Theoretical Population Genetics, and there are also more involved ones by a mathematician, Duret. On this part, you could look up the book by Bürger. Okay.
And stop here.
Yeah.
And